Yeah, it's my pleasure to introduce Professor Squire Booker from Penn State, who will tell us about crystallographic snapshots of TsrM, a Cobalamin-Dependent radical S-adenosylmethionine methylase. Thanks.
Thanks so much for the kind invitation. I'm really excited to participate in such an important endeavor, especially with the PDB being 50 years old now. So my laboratory for quite some time has been interested in methylation reactions catalyzed by S-adenosylmethionine dependent enzymes. And so S-adenosylmethionine has been called nature's universal methylating agent. It's the source of the vast majority of methyl groups that are appended to a wide variety of substrates, including proteins, carbohydrates, lipids, DNA, RNA, and various small molecules. Traditional SAM-dependent methyltransferases operate by an SN2 mechanism wherein a nucleophile attacks the activated methyl group of SAM in a polar fashion to give a transition state that looks something like this, which then collapses to your methylated product, shown here, and the co-product, S-adenosyl homocysteine. Now, using this very simple strategy, a variety of biological nucleophiles are known to acquire methyl groups from SAM, including nitrogen, oxygen, sulfur, arsenic, selenium, and tellurium. And you can even methylate carbons. However, that can only be done if you're able to generate a suitably nucleophilic carbanion, as is seen in the DNA methyltransferases. Within the last 20 or 30 years or so, it has become apparent that non-nucleophilic carbon atoms can also receive methyl groups from SAM. But it's only in the last 10 years that we've begun to understand the mechanisms by which these methylations take place. And so on this slide right here, I'm showing you a variety of natural products that contain inert carbon atoms that become methylated during their biosynthesis. And so here's phosphomycin right here. Here's a precursor to thionomycin, an antibiotic. Here's bacterial chlorophyll, et cetera, et cetera. In all instances, the enzymes that append methyl groups to these inert carbon centers belong to the superfamily of enzymes called radical S-adenosylmethionine. And radical S-adenosylmethionine enzymes all contain at least one cofactor called a 4-iron 4-sulfur cluster, shown right here, that is ligated by 3 cysteine residues. The 4th iron of this cubane cluster has an available coordination site to which S-adenosylmethionine itself can coordinate in a bidentate fashion through its alpha amino and alpha carboxylate groups. When this cluster receives an external electron, it can induce a fragmentation of this C5' carbon sulfur bond to give this highly potent 5' deoxyadenosyl radical. So we have an unpaired electron on a primary carbon. And the role of this radical in this vast number of enzymes is simply to abstract the hydrogen atom from an enzyme-bound substrate. So several such radical SAM methylases are involved in the biosynthesis of carbapenem antibiotics. And carbapenems are antibiotics of last resort in the clinic and are increasingly important in the fight against multi-drug resistant bacteria. They are highly broad spectrum antibiotics containing this central beta-lactam core shown right here. And it's this beta-lactam core that is responsible for their bactericidal activity through the inhibition of cell wall biosynthesis. Importantly, This C6, this is the sixth position, hydroxyethyl group in thionomycin, or the C6 hydroxypropyl group in asparagomycin, distinguishes these clinically used carbapenems from other known classes of beta-lactam antibiotics. And these side chains actually prevent these antibiotics from being inactivated by beta-lactamases. And so thionomycin, shown here on the left, is one of the most potently naturally produced antibiotics known to date. And it exhibits potent activity against both gram-positive and gram-negative bacteria. On the right, I'm showing you asparinomycin A, which also exhibits potent broad-spectrum antibiotic activity. Now, both thionomycin and asparagomycin A are synthesized by similar pathways. In thionomycin, there are two sequential methylations that give rise to this hydroxyethyl C6 side chain. And in asparinomycin, there are three sequential, 1, 2, 3 methylations that give rise to this hydroxypropyl C6 side chain. Importantly, in both of these biosynthetic pathways, there's one single enzyme that catalyzes both of the methylations in this particular case and all three of the methylations in this particular case, okay? So methylcobalamin is the central cofactor in the Tokk reaction, which is the reaction that I'll talk about today. And so here's the structure of methylcobalamin shown right here. Here's the cobalamin core and ring. Here's the dimethylbenzimidazole tail that is sometimes coordinated to the cobalt ion. And of course, here's the methyl group of methylcobalamin. And so the cobalt ion can exist in three distinct oxidation states. When you have alkyl cobalamins, the cobalt is in the +3 oxidation state. And it's almost always 6 coordinate, meaning that there's something bound to this bottom face of the cobalt ion. If you displace the methyl group, you arrive at the COB1 oxidation state, where the cobalamin has a +1 charge, and COB1 element is almost always 4 coordinate. And then there's the intermediate oxidation state, which is COB2 element, which has a single unpaired electron in the DZ² orbital. COB2 element is paramagnetic, and it can exist both in a uncoordinated and a coordinated state, as shown right here. Now, the general mechanism for these cobalamin-dependent radical SAM methylases is shown on this particular slide. And this is the mechanism expected for Tokk. So here's our iron sulfur cluster to which S-adenosylmethionine is bound. When that cluster gets an electron, it promotes the cleavage of S-adenosylmethionine to give this 5' deoxyadenosine radical. This radical can then abstract stereoselectively a hydrogen atom from the C6 position of the carbapenem substrate to give 5' deoxyadenosine, one of the products, and this substrate radical shown right here. In a subsequent step, this substrate radical can then attack in a single electron nucleophilic mechanism the methyl group of methylcobalamin, and that causes this bond right here to fragment homolytically. So one electron will pair up with the electron on the substrate, and the other electron goes to COB3 element to make COB2 element. So that gives you your methylated substrate now. Your COB2 element picks up another electron from an external source. That gives you COB1 element, which is a supernucleophile, the most nucleophilic biological molecule. And then COB1 one alderman does a standard SN2 reaction on SAM to regenerate methylcobalamin and kick off S-adenosylmethionine. And so Tokk will do this reaction 2 additional times to generate this propyl side chain shown right here. And so the stoichiometry of this particular reaction is that 2 S-adenosylmethionines plus your substrate gives rise to 1 5'-deoxyadenosine shown right here. And the co-product of that is methionine, plus your product shown right here, and then plus S-adenosylhomocysteine, which is formed when the cobalamin gets remethylated by SAM, okay? Now, this project is a collaboration between Craig Townsend's laboratory and his student, Erica Center. And Erica did a time course, and her time course indicates that Tokk installs the second methyl group more efficiently than the first, while the third is installed the slowest. So this methylation on your substrate takes place with this rate constant, which is very slow, to give you your first product. But then the second methylation takes place somewhat faster than the first, and the third is the slowest of them all. And this is a bit counterintuitive because one would think that the first methylation should be the fastest, because the radical that you would generate is on, first of all, a secondary carbon, and secondly, the radical is stabilized through resonance into the carbonyl portion of the cofactor of your substrate. By contrast, if you look at the second methylation, you're abstracting a hydrogen atom from primary carbon, which is energetically unfavorable. And so Haley Knox, a graduate student in my laboratory, solved the structure of Tokk in complex with 5'-deoxyadenosine and methionine, which are the cleavage products of S-adenosylmethionine. And this is the structure shown right here. It has 3 domains. The central domain right here is the radical SAM domain. It's a shortened alpha beta 6 TIM barrel. And the radical SAM domain houses the iron sulfur cluster, and it's where S-adenosylmethionine binds. A second domain here is a Ross manifold that binds the cobalamin cofactor. And then there's this third domain, which participates in substrate bonding. And so this is sort of what the active side of the enzyme looks like. Here's your cobalamin cofactor. Here's your 5'-deoxyadenosine. And after cleavage, the methionine remains coordinated to that unique iron of the iron sulfur cluster. Just underneath the cobalamin, cobalt ion of the cobalamin, is this tryptophan residue, which we believe serves to maintain the cobalamin in a 5-coordinate or 4-coordinate state by preventing access by water or any other small molecules. Now, when we solve the structure of the enzyme in the presence of this carbapenem substrate, which I've shown you right here, we begin to get answers about why the rate constants are counterintuitive. So the C6 carbon of the substrate is about 3.8 angstroms away from the 5' carbon of 5'-deoxyadenosine. So this is where you would generate a radical, and that radical would abstract one of the hydrogen atoms from carbon 6 right here. In addition, the angle of this 5' carbon and the C6 carbon is not ideal for hydrogen atom abstraction. And so we think that substrate positioning probably prevents methylation due to difficulty in abstracting the first hydrogen atom and then putting the methyl group onto cobalamin, shown right here. We can project what we can project where we believe the methyl group will be appended. And if we look at the distances from the 5' carbon and the methyl group of the cobalamin, those distances seem to be much more favorable for hydrogen atom abstraction from the methyl group, as well as subsequent methylation of the substrate from methylcobalamin. So the enzyme that I just talked to you about, Tokk is responsible for methylating a carbon that is sp3 hybridized. However, these cobalamin-dependent radical SAM enzymes can also methylate carbons that are sp2 hybridized, as in the case of TsrM that I'm showing you right here. So TsrM catalyzes the first step in the biosynthesis of the quinaldic acid moiety right here in this antibiotic right here called thiostripton, all right? In contrast to Tokk, TsrM is using SAM to put a methyl group onto an sp2 hybridized carbon, shown right there. Now, interestingly, TsrM, although it's a radical SAM enzyme, it doesn't catalyze formation of 5' deoxyadenosine. So if you run the TsrM reaction, you see your methyltryptophan product, you see S-adenosylmethionine, but you don't see 5' deoxyadenosine, which would come down here. This is in contrast to the Tokk reaction, where you see 5' deoxyadenosine methionine and S-adenosylmethionine. So why does the radical SAM enzyme catalyze a reaction that doesn't cleave SAM to generate a 5' deoxyadenylyl radical? So Haley Knox solved the structure of TsrM as well. Similarly to Tokk, the cobalamin is bound in this Rossmann fold right here. This in silver is the radical SAM alpha-6 beta-6 TIM barrel, and then in purple is a similar C-terminal domain that helps to bind substrate. The interesting thing about this structure, when we took a look at it, is that the iron sulfur cluster is fully ligated, in contrast to every other radical SAM enzyme that had been studied. Here are 3 cysteines to 3 irons of the cluster, but the 4th iron is ligated by a glutamate residue. Also interestingly, there's an arginine that sits just under the cobalt ion of the cobalamin cofactor. So this glutamyl residue that ligates to the iron sulfur cluster prevents SAM from ligating to the cluster, which also would prevent SAM from being cleaved to generate a radical. We also solved the structure in the presence of a SAM analog called azosam, where there's nitrogen in place of the central sulfur atom, and the substrate tryptophan. But if you look at the substrate tryptophan, the carbon that is to receive a methyl group from methylcobalamin is displaced and is not bound in a conformation that should allow its methylation. And so we did some computational docking studies and found 2 poses of almost equal energy, shown right here. One of those poses completely recapitulates what we see in the X-ray crystal structure, that's pose 1. But pose 2, shown right here, puts the carbon of C2 carbon of tryptophan directly over the methyl group of methylcobalamin, but also the nitrogen of tryptophan is in hydrogen bonding distance to the carboxylate of S-adenosylmethionine. And so these are 2 poses of that exact same structure. All right, and so the question that we want to ask now is that, is bound SAM required for methylation of tryptophan? And so we're going to do this following experiment. We're going to treat TsrM with S-adenosylmethionine under reductive conditions to generate the methylated form of the cofactor. So we're going to generate methylcobalamin on the enzyme. We're going to separate the enzyme from all the small molecules by gel filtration chromatography. And so here's TsrM that has methylcobalamin bound to it. And now we're going to take the methylcobalamin bound TsrM and treat it with analogs of S-adenosylmethionine in the presence of tryptophan. If we have no S-adenosylmethionine, as shown down here in black, we see no methylation of tryptophan, meaning that S-adenosylmethionine needs to be present. If we have S-adenosyl homocysteine, which is an analog of S-adenosylmethionine, we get some methylation. But if we have ACA, which is a better analog of S-adenosylmethionine because it recapitulates the positive charge of the sulfonium group, we get even better production of methyltryptophan.
Just a minute or so.
Okay, I'm just about done. So here's the structure of decarboxylated SAM, and the question here is the carboxyl group of S-adenosylmethionine important in the in methylation of tryptophan by TsrM? And shown here, what we're looking at is that methylcobalamin-bound enzyme in the presence of decarboxylated SAM, and you see no activity whatsoever. There's just a very small amount of residual activity in the presence of 50 micromolar enzyme. By contrast, on this in this panel, we're showing that in the presence of decarboxylated SAM, the decarboxylated SAM can actually methylate the cofactor of TsrM to give rise to methylcobalamin. So decarboxylated SAM functions just fine to methylate cobalamin. However, it does not allow formation of your methyltryp product. And so lastly, I'll leave you with this mechanism for how we think this reaction takes place. So here's the carboxylate of SAM that's acting to receive a proton from tryptophan during an electrophilic addition mechanism, all right? So the electrons from nitrogen will swing in up to C2 and attack the methyl group of methylcobalamin. From this particular intermediate, you lose that proton. The proton from SAM goes back, and you get your methylated product. I'll dispense with all the other stuff in the interest of time, and just thank all the people that have worked on the project. Haley, we worked in collaboration with Kathy Drennan's lab and her student, Percy Chen, and thank you for your attention.
Thank you, Squire, for that beautiful talk. In our increasingly antibiotic-resistant world, this kind of detailed structure function analysis is, yeah, it's beautiful. I'm sure that Squire would be happy to answer any questions. I have a question about the beta-lactam ring, which, you know, there's been suggestion that it's that strain in that ring, the non-planarity, can actually lead to differential reactivities. And I'm wondering if you think that plays a role here.
So that's a good question. It's not something that we have thought about. So, you know, I can understand how the strain in the ring would lead to differential reactivities with, you know, the enzymes. You know, it inhibits these penicillin binding proteins. And I can see where that would be important. In this particular case, I'm not convinced that it has anything to do with methyl groups, you know, being appended to C6 of the antibody.
Any additional questions?
Okay, well, oops, thank you.
Yeah, thank you very much for that fantastic talk. I'm sure Squire would be happy to answer questions in the chat.